What I want to do with you today is to give you the information about lipids, to give you lipid information about proteins. We already talked about enzymes today, and then also hit nucleic acids. On your calendar, tomorrow was supposed to be the test for this chapter. Let's push it to Friday. Okay? But listen, guys, I'm sorry. The problem is that backs us up for the next chapter, okay? You have to understand, there is no break, right? So if we push, this is not your fault, it's mine, because the materials weren't here for us to do all this preparation yesterday for us to be ready today to do this lab, okay? This is fine. We hadn't gotten through all the material yet anyway. So therefore, today I want to get through this, and I'm going to have to go reasonably quick, okay? And again, the study guide is your key. And then tomorrow, we'll hit the lab. I think that we can finish that. It says we can do it in a 15-minute block. And I think having you broken down into the pieces, making sure everyone in your group is doing something, right? So in other words, Nathan's not just sitting there staring lovingly at someone in his group. He's actually working the whole time, right? Okay? And that wasn't meant to be. And Tony's not just staring up at the ceiling and wondering and pondering the meaning of life, right? I mean, like we're actually working. Are we cool? Sorry, I'm just you know me, I just grab people. All right, are you ready? So let's dive in. 4 pieces, right? Proteins, lipids, carbohydrates, and nucleic acids. Those are our 4 main biological molecules. We hit carbohydrates in significant detail yesterday. Let's talk about lipids. Lipids are fats, right? If you look on your study guides, you see that there are a couple of pieces that I want you to know about lipids in general. They are also made of carbon, hydrogen, oxygen, but a much higher ratio of hydrogen to oxygen. We talked about this yesterday. More calories per gram. In other words, fats or lipids, if you eat the same amount as a protein or as a carbohydrate, it has more calories. They're denser, right? And also, their primary function is to do what? To store energy. Now again, they have lots of other functions. We'll talk about some of the other pieces they do. But this is the primary function I want you to be comfortable with. So let's talk about these fatty acids. Excuse me. They are chains of carbons. At one end they have a carboxyl group. At the other end they have a methyl group. And if you remember that there are these different pieces. If you flip back onto page 56, they show you the names, right, for these little pieces that are attached on these functional groups for these organic molecules. Okay, again, you don't have to know them, but just when I toss out this carboxyl, you know what I'm talking about. Okay? Sorry, I'm trying to chew my gum and talk at the same time. It doesn't work very well. And a key piece between these guys, because I'm sure you've heard of things called saturated fats or unsaturated fats. If a fat is saturated, if a fatty acid is saturated, it means that every single bond, every single carbon is full of hydrogens. Okay? And if it's unsaturated, apparently it means that there are some bonds that are not full of hydrogens. That they're bonded to something else, or this is simply hanging out there on its own. So, nice saturated fat, right? All H's, H's, H's. And then, oops, wait a minute, this guy right here, there are no H's there, so there's a double bond between the carbons. It's sharing 2 electrons instead of only 1 each. Does that make sense? That's a key piece between those fatty acids. And those are digested differently. They can make them, whether they are oils or fats, whether they're solid or liquids, their structure determines their functions as well as their characteristics. Okay? We can, of course, take these fatty acids and combine them together, right, to make larger fats, right? So that's these trans fats or these cis fatty acids. That deals with the shape of how they are actually bonded together. And those trans fats are a little bit harder for our body to digest. They tend to build up a little bit more. Your textbook talks about them being bad, though there's some research that is coming out now saying, well, maybe trans fats aren't this terrible thing we've talked about. We're actually in some ways doing... Anyway, there's a whole research about this. There's no easy, good answer. Oh, trans fats are so terrible. The FDA even banned them in 2015. Said you can't, in a restaurant, you cannot serve things that have trans fats in them. Okay, so there's certainly some difficulties there. And again, the point of that, the why is because they were building up. And the thought is that helps to cause heart disease and problems.
Isn't it? It's like it breaks down properly.
Right, it won't digest as easily. Yeah, sir.
Say if you're at home, right, and you cook something from scratch, how are you gonna know if it has like?
Well, depending on what you're actually frying it in, makes a difference. The oils that you're actually using, you can find out from those pieces. So the ingredients you put in, most of them, again, at this point, they're not selling it with those. There are some, yes, unfortunately. But again, that's why there's some research, there's some controversy as to whether this is really as bad as they make it seem. Does that make sense? It's like it just kind of, everybody was against trans fats, and so it was to the point that we banned them totally. But are they really that bad? Well, there's some back and forths.
What was that piece about good and bad?
Cholesterol, we'll come to that. That's the steroid pieces, yep. For lipids, our next piece is phospholipids. And these you should be very familiar with from your time in biology, right? What do phospholipids do? They make up a huge role in your body. The cell membrane, the outside wall of the cell, is made up of phospholipids. Because they have this amazing relationship, their heads, the top of them is a phosphate, and it's hydrophilic. It's hydrophilic, it loves water, right? Whereas the tail, the end of it, the fatty acid side, is hydrophobic. So therefore, it sets up this membrane where the outside keeps water from coming in and the inside allows it to. It allows a barrier to let some things in and some things not, depending on how they're moving and where they're going. So if you put up a phospholipid bilayer, maybe you've heard this term before in your biology studies, right? That you are allowing some things to enter the cell, some things not. And it provides structure to the cell. Does that make sense? All of our cells, I can't think of any one that's not. The cell membrane is this phospholipid bilayer. If we continue on, you've got hormones, which I kind of skipped past there because I'm not too worried. We'll talk more about these in the future. We have our steroids that are basically, you have 4 rings of carbon that are together. They look something like this, right? So here, these are carbon rings, and each of these points is a carbon with, again, these little pieces stuck onto the ends. And how they stick on determines what actual substance it is. What do these steroids do? Well, cholesterol is like the parent one. And what this does is it allows your body to do things. Steroids are hormones, right? So hormones belong to what system? What system in your body uses hormones? Endocrine, right? I'm making you recall stuff you've learned, right? So these are the messengers that are sent out that at puberty tells a young man, hey, start growing chest hair, start voice getting deeper, start actually being able to produce sperm. To the woman, these steroids come out, these hormones tell them, okay, develop breasts, have the ovaries start producing eggs, start a menstruation cycle. Does that make sense? They're hormones, okay? Hopefully not. There are lots of other ones too. Cortisol is a stress hormone. Progesterone, estrogen, testosterone, those are all sex hormones. Bile acids is actually from your liver that's secreted into your intestines to help digest fats. We'll talk more about these pieces. That's it for cholesterol. There's HDL and LDL cholesterol. I didn't put on your study guide. I'm not really interested in us spending too much time. The key piece I want you to understand is fat is not bad. Fat is given a terrible name in America. eating-wise, okay? You need fats. We've just gone through 5 or 6 different ways your body actually uses fats, right? Consistently. So if you say, oh, I'm having a fat-free diet, well, it's really not the best plan, right? You need some in order for it to work. Okay? And the key here is you would like to have more HDL than LDLs. Sir? Mm-hmm. Right, exactly. Fat by itself will not make you obese. You're correct. Yeah, certainly. Okay, let's talk about proteins. Proteins are polymers, right? What is the monomer of a protein? An amino acid. These are the alphabet that make up the words. Proteins, the words, amino acids, the alphabet. Are you with me? There are 20 amino acids that we use, and those in any combination will make all the proteins in your body. Just simply 20. That's it. I'm not giving you per se that, but it's a good thing to be comfortable with. You can actually, it's on your study guide, so I would be comfortable with that number. Sure. Oh, no, my word, no. Oh, no. No. And then you also have an R group that's added on. So again, it's very similar to those carboxyl, the pieces that are added on, that it helps provide a specific function. Here are your amino acids. So again, these are the same, these pieces are the same, but the R group is different. This is the same, the R group is different. And these are specific amino acids. Methionine, cysteine, triosine. These are the names. Actually, all 20 of them have names. Again, the further you go, you will have to memorize all 20 of them and what they do and how they work. But
so the R groups are part of the amino acids?
They are.
That's the only thing that makes them different from each other?
Exactly, because this base piece is always the same.
Okay.
Yep. Okay, the key piece about proteins is when I take 2 amino acids and put them together, there's a bond that forms between them, right? Again, think about our monosaccharides becoming disaccharides, right? Same process, except we give it a name. We call that bond between 2 amino acids a peptide bond. Okay? It is bonding 2 amino acids together. Cool? Does anyone know where that happens in the cell? Do you remember? What part of the cell does protein synthesis? RNA is a part of it. Not the mitochondria. That's the powerhouse of the cell where we do aerobic respiration. The ribosome. Ribosome. We'll talk all about that. Okay, so again, here's just showing you peptide bonds. These 2 amino acids come together. They form a bond. Water comes out. Do you remember what we called that yesterday when we formed a bond and water came out? Dehydration synthesis. We're taking out a water and synthesizing a new molecule. Let's keep going. Conformation. This is a huge thing in proteins. I don't know if your textbook has a picture of this. Oh, there you go. Yes, page 65. On page 65, you see a picture that looks like this. Are you comfortable that amino acids are just a chain, right? These peptide bonds make a chain, right? This polymer from a bunch of monomers. And that chain is just what we call the primary structure. And again, you can actually take a protein and sequence its amino acids to say, if I just give them numbers, that the sequence is 2, then 7, then 15, then 23, then 6, then 5, then 4. And that's your chain of amino acids that are all bonded together, right? And every protein has its specific order. Just like if I want to spell the word Alexander, I have to have specific letters in a certain order, right? If I want to spell the word autumn, I have to have specific letters in specific orders. If I want to spell Carlos, right? I got specific letters in a specific order. Amino acids are the same. Each protein is just a word. It's a very long word, by the way. Lots of letters. So that's our primary structure. Then, this chain goes through this three-dimensional folding process. Through these bonds, through hydrogen bonds, through the van der Waals forces that are bonding these things together. It then turns into, again, it's still a chain, right? But it now has this three-dimensional structure that they call the quaternary structure. By the way, this is just simply Latin for 1, 2, 3, 4. Primary, secondary, tertiary, quaternary. 1, 2, 3, 4, that's all that means, so don't freak out about that. The point is, this three-dimensional shape determines function. Let me say that again. The three-dimensional shape of a protein determines its function. That's why if I take the egg, right, and I fry it up, I've changed the structure. It can't go back to what it was. Are you with me? If I take that enzyme and I put it in stomach acid, it's going to denature it, right? It's going to break it down. It's going to change its shape. So therefore, it won't work anymore. Does that make sense? I always tell this story when I come to this. Do you all remember Mikey and JoJo Brown and Tyler Brown? Coach Brown, he did basketball for a good while. Milton Brown, right? So he works down at Georgetown. I told you this before. He runs a lab down there where they design drugs. And I took some chemistry students down there. This must have been about 6 or 7 years ago at this point now. We went down there. And one of the things he was so proud of showing us was this computer where if you wore these 3D glasses, you could see specific proteins that they had modeled on this computer, and you can see it in three-dimensionality and turn it and look at it. And this was a very cool thing because that's what their work is, right? Are you understanding that to build a drug, you simply have to make a protein or make a compound that will fit, like you're building the lock that will unlock this key will unlock. Does that make sense? To basically shut down a virus or shut down a bacteria or to take your body and tell it to do specific things. Are we cool? And so he was very excited about showing us this, and we all got to put the glasses on. You really couldn't see it all that great, but okay, it was fun. Now, all of those three-dimensional structures are on a database on the web. Like I could just go there right now and we could pull up, we can look at, here is the protein structure, the three-dimensional structure for this certain thing. Like that's how far computers have progressed since then. It hasn't been that long since we did this, right? My point is just simply this. This is seriously important. The conformation process, that's what this is called. Folding these structures into specific shapes, because shape determines function for protein. Have I said that enough now? Shape determines function, okay? Because protein has so many functions, right? It does hundreds of different things in our bodies. Just a couple of them that we mentioned there. Nope, let's keep going. Nope. There we go. Structure, communication, membrane transport, catalyst. Enzymes are proteins, right? So let's go through a couple of these. Structure, the proteins in our skin, the proteins in our hair, the proteins in our teeth, in our cartilage. All of these are different, right? They all play little different roles. Communication. Some hormones are proteins, and some of the signal cells that they use to actually communicate with nerves are proteins. Membrane transport. We've got these little channels in the cell membrane, like little gates that allow things to come in and out. And again, this is a protein. Are you with me? Tons of different functions for proteins. The whole process of your immune system is basically recognizing the proteins on the surface of bad things and killing them. I know this is in health class we're talking about this. But it's basically like a dog coming up and sniffing, like, do I know you? Oh, okay, I know you, you're good, and I'll leave you alone. That's what antibodies do. And how they do that is by looking at the proteins. They basically check out the surface of this thing. If it has bad proteins, because it's a virus or a bacteria, boom, kills it.
Virus is
excuse me, they fight back by changing very quickly. So they don't really have active defenses. Like we have whole series of cells that fight off these bad things. They don't really have that same thing. Their key is that they want to come and take over a cell and then use that cell to make more of itself. And they'll change very quickly. They can what's the word I want? Mutate very quickly. Yeah, that's their primary piece. Are you with me, my friends? About proteins. Let's talk about enzymes. I mentioned it in our lab talking about this earlier. Again, these are biological catalysts. The substrate is what the enzyme is acting on. What does the enzyme produce? That's what we call a product, right? And the key thing, we talked about this before, that enzymes lower activation energy. It makes it easier for the actual reaction to occur. Okay? That's the key piece with enzymes. And you remember that ACE, right? ACE will clue you in that we actually have. So this is just kind of showing you a graph, right? If I don't have a catalyst, I need to get this certain amount of energy before I can start the reaction. Whereas for using an enzyme or a catalyst, it lowers that activation, makes it easier for the reaction to start and easier for it to continue. Are you cool? Yes, ma'am.
So are enzymes just like chemicals or like?
They're proteins.
Oh, they're
They're proteins. Yep, they are proteins. Okay? We've already talked about all of this stuff in the lab. So I don't feel a need. Here's your textbook showing you how this works, right? In this case, again, they are showing you a decomposition reaction. Here's our substrate coming in. The enzyme bonds, and so you have this thing they call the enzyme substrate complex, which just means the two things are one now, right? The bond is broken between those two pieces of the substrate, and you have products that are individual that are released. Are you comfortable some enzymes do the opposite of this? That they take let's take two monosaccharides, and they bring them together, and they bond and form a disaccharide? Are you with me? Cool. This is how we make glycogen. It's how we store. Good. Enzymes are reusable. They're not used up in the reactions. They just are playing a role. They're just helping it along. They do work super fast. One enzyme molecule can consume millions of substrate molecules per minute, which is impressive. It's a factory. Boom, it just gets this stuff out. Pumps them out, because we need them, right? We need these things quickly. And we can affect enzyme shape and function by temperature and pH, which of course that's what our lab is showing. Any questions there? Good, stand up. Let's get you moving a little bit. Stretching. Whoa. Because we're almost there. We're almost there. You're good? 17 minutes. It goes quick, right? Man. I'm trying to be aware. I don't want to keep you sitting because I know you get tired. I know you get tired. Maybe we should go like through the volleyball warm-ups. We'll do some conditioning here, right? That'd be excellent. All right? Okay, we'll get the hurdles, we'll get the ladders. No? Would be fun. Would be fun. All right, good. You're welcome to sit back down again. All right. So, what was the monomer for a carbohydrate? The monomer for a carbohydrate. Okay, glucose is one of them. There's a name for those three. Monosaccharides, right? Those are the building blocks. You've heard that maybe in your previous classes. The building blocks of a protein are what? Amino acids. Those, again, are the monomers of a amino acid. Fats, you may have noticed we skipped. I didn't give you a monomer of those because there's really debate as to what is the monomer. Whether it's the fatty acid is really kind of the basic, but I won't bore you with all that pieces. There is no good solid monomer for lipids. But the monomer for nucleic acids is what? A nucleotide. Okay? These are like easy questions on a test, right? Because you will see things like that. So, nucleotides are the monomers. And they have three pieces. They have a nitrogen base, right? Along with carbon. They have a sugar of some sort, a monosaccharide. Do you know the only difference between DNA and RNA? The sugar in the molecule. That's it. One is called deoxyribose. The other one is called ribose. That's it. These are nucleotides. Yep, the three parts of a nucleotide is the base, the sugar, and then one or more phosphate groups, right? We'll show you the picture of that. And again, ATP is the best known nucleotide. And again, it comes from adenosine triphosphate. So how many phosphates are on an ATP? 3. And if it breaks down and loses one of those P's, what does it become? Adenosine di, which we call a DP. And you realize that's how your body does all day long. It takes ATPs, breaks off one P, and uses that energy for your body to work. Right? It then takes that ADP and sends it back to the ribosome and stores more energy in a bond with that other P and turns it back into ATP. And the cycle continues. Okay, that's 2 chapters from now. We'll talk more about that in significant detail. Okay, so here's again a view of ATP. Okay, with the phosphates, with the sugar. Let's keep going. Yeah, we just finished talking about this. Good. Because this ATP, and I'll say this again, as I wrote on your study guide, it is the body's most important method of transferring energy. Okay? We store it. Where do we get our energy from? Our food. Good. All of our food is broken down really into one main molecule. Do you remember? Carbon is good. Good. That would just be an element, though. A carbohydrate, good. In particular, what carbohydrate? Glucose. Everything breaks down into glucose. I'm generalizing. There are some things that don't. But the food we eat that we're going to use to make energy, we break it down into glucose. That glucose then is converted through the aerobic respiratory process, which we'll talk all about. The products that come out are carbon dioxide and water, right? All the water we need to actually live. The carbon dioxide we breathe out, the waste product. And that is then converted from ADP, we change ADPs into ATPs. And those ATPs can be used for muscular contractions, for cilia moving, for active transport, for synthesis reactions, for anything. All the stuff the body does needs ATP. Does that make sense? And again, how does that work? This last P is broken off. That last P comes off, and that gives energy. It releases the energy that's been stored there. Then ATP goes, and they rebond a P to that ADP and turn it back into ATP to store more energy, and the cycle continues. Question?
So ATP is basically what's left from glucose when it's been...
Yes. You could say that. Yes. It's a little simplified, but yes, I'm comfortable with that. The whole point of taking that glucose is we're going to burn it. We're going to use it and break it down and make ATPs. Yep. Are you with me, my friends? And again, that all happens in the mitochondria. We'll talk a whole bunch more about this, where we're actually breaking down the glucose into two other pieces, into pyruvate, and then it goes through this. You can have no oxygen present, and it can turn into lactate. You can actually use the mitochondria to break it down. We're going to have to spend plenty of time on this in the future, so don't worry about that piece. The point is, we're taking glucose, and we're breaking it and making ATPs. I've given you more information about nucleotides. So if I take nucleotides, which again, those are the monomers, right? The monomers of nucleic acids are nucleotides. If I put a bunch of those nucleotides together, I get a nucleic acid. You with me? If I put a bunch of monomers for carbohydrates together, that would be a bunch of monosaccharides turning into polysaccharides, right? If I put a bunch of monomers of proteins together, that would be amino acids turning into proteins, right? Same process. A bunch of little pieces, nucleotides, come together to make a nucleic acid. The two big nucleic acids you should already be familiar with, DNA and RNA. DNA, of course, constitutes our genes. It stores all the information that makes you you. The exact code that makes you you. And all the proteins and all the organs and your specific lips and your specific eyes and your specific ears and how tall you are and how well you digest pizza. That's all covered by that. Okay? And RNA is like the messenger, right? That's one of them was messenger RNA, right? Where it actually transfers pieces of that DNA code outside of the nucleus. You've got ribosomal RNA, which is actually helping the RNAs, the DNAs to work. We'll talk much more about these pieces. One of the key things is it makes proteins. That process of making new proteins comes from RNA. Any questions? It's a good bit of stuff. It's a good bit of stuff. And again, we're glancing the surface of pieces that I want you to make sure you know for our test on Friday. But do check out the textbook. I'm assuming you are reading, right? You've noticed I've not said, hey, Abby, you got to read chapter 2. I'm assuming you have, right? Yep. Yep. It's just a cycle. Yep. Glucose, the energy that we get from glucose is stored in the bond between that last phosphate when it becomes ATP. So it has two phosphates, ADP. We put a third one on there, and now it's ATP. We take it to the cell. So these guys here are the ribosome, okay? And they are breaking down glucose, not ribosome, the mitochondria. They're breaking down glucose and turning ATP into ADP. Sorry, other way, man. ADP into ATP. Okay, so they break down the glucose and there's energy that's released. Give me some energy. Give me some energy right here. And I take a P and I capture that energy. So now I have ATP, right? I'm going to take this ATP over here because this is a bunch of muscles here, right? They need to work. Okay? You're going to break off that P, release this energy, you've got power and strength now. But now I'm just an ADP, so I'm going to come back over to my mitochondria. Give me some more energy. Phosphorus, boom, now, okay, I got another phosphate, now I'm ATP again. Does that make sense? And we do this over and over and over, millions and billions of times in our body. We cool? All right. I'm going to stop this here.